Okay, let's get started. Couple of quick announcements. The first one is, as I think I've mentioned before, I'm not going to be here on Friday because I'll be at the University of Florida, which means that we will have a guest lecture. That will be Dr. Mike Bartlett, currently chair of the biology department here. He's an expert on transcription, and that's what he's going to be talking about. I don't think he will be recording his lecture. So if you have recordings you'd like to do that, recommend that you do so. There are probably not going to be clicker questions. I haven't asked him yet, but I suspect that will be the case. There will not be clicker questions. So no clicker questions. Why do you come? Because it's not going to be recorded. So that being said, I also have posted a couple of hints, tips, tricks for doing well on exams on D2L in the discussion section. So if you're interested in how other people have done well, at least on this past exam, I suggest taking a look at those. I do have your Scantrons if you want to pick them up. I'll be setting them out after class over there on the side. They're not there yet. The last announcement, at least for now, you'll probably see this a whole bunch. Up here in the middle, most of you probably heard about virology next term, which will be Monday, Wednesday, Friday, 9 to 10:05. Not here. We'll see where they end up scheduling us, and that will be me. I'll talk a little bit about viruses from hell there. The thing that you may not have heard about is the, what are we calling it again? The Advanced Molecular and Cell Biology Research Lab, also known as Mutant Viruses from Hell. This is sort of the virology lab that we are doing here. By we, I mostly mean me and a couple of my graduate students. Basically, it's a real research course. You start out with a couple of mutant viruses from hell, things that we work with in my lab. We have no idea what those mutants are. And then over the next 10 weeks, you tell us what those mutants are and how cool they are. And that, yeah, it does take 10 weeks, believe it or not. So there are going to be two sections. It's Monday, Wednesday afternoons, and Tuesday, Thursday afternoons from, and then what do we say, 1 to almost 4. Rarely takes us that long, but sometimes it does. And then the very last thing, not advertised anywhere, you're hearing it here first, there's going to be a 1-credit seminar class on Fridays about reading literature and presenting and discussing literature. So I will give you more information about that as time goes by. Why am I telling you this? Because some people at least can sign up for classes for next term. Like, next term already? Seems way too early. Okay, any questions about that before we move on to the rest of today's lecture? No. Okay, so today we're going to continue talking about DNA repair, and just as a review, the main things that we, as organisms living in an oxygenic environment, or not oxygenic environment, an oxygen-containing environment at a relatively high temperature, and we like light. Again, this is a nasty thing outside today. These are the main things that are issues as far as DNA is concerned. And the changes which happen to DNA, the chemical changes that happen to DNA, which then, of course, can lead to mutations. So what do we mean by mutations? Okay, we talked about the chemical changes last time, but there's also some very important terminology here that I wanted to go over. There really are two major kinds of mutations that are going to be transferred genetically, either transitions or transversions. And this allows me to introduce some of this ridiculous single-letter code for nucleotides. So Y stands for pyrimidines, because there's a Y in pyrimidines, and R stands for purines. So any pyrimidine, which would be what? T's or C's, and any purine, which is going to be A's or G's. Exactly. So if you have a transition, that's going to be from pyrimidine to pyrimidine or purine to purine. And if you think about that from the structure of DNA, which all of you, of course, remember, since we covered it in the last midterm, purines and pyrimidines are going to have different base pairing, but they're going to be pretty similar sizes. And so they'll fit relatively well into the DNA. A transversion, is, on the other hand, is where you go from either small to big, so pyrimidine to purine, or big to small, purine to pyrimidine. And that, of course, is going to change a lot the structure of DNA, because it's going to be very different in terms of the space between the two different strands. And so that's going to be a very obvious kind of difference that you have here. It turns out that transitions also give a slight change in terms of the structure because the base pairing is not working anymore. But clearly, the transversions are going to give you much more of an issue. And that's what's supposed to be shown down here at the bottom. Here we have so-called mismatch repair. This is a process which happens not because we like it warm, we like it light, and we like oxygen. This is just because DNA polymerases occasionally will make mistakes. And sometimes those mistakes are going to get, when I say mistake, just the incorrect nucleotide gets added in the new strand or your non-template strand. Usually that'll get recognized by what? Exonuclease proofreading activity, but sometimes it doesn't. You remember, these polymerases are chugging along at hundreds of thousands of nucleotides a second, so they're really fast. So occasionally you get the wrong nucleotide which is added, and the way that's dealt with is what's called mismatch repair. Mismatch, you've got two bases that are not properly base pairing with each other, so you need to repair them. And this is the mismatch repair process. And we'll talk more about that as we move on through the rest of today. So I like to think of it as a repair process. Often it's covered as part of replication. Yes? So the question is, it would only check for transversions. It turns out it will also repair transitions, just because the base pairs are, the hydrogen bonds are off enough that change, and it's a very, very, very specific measurement of the structure, that will also get detected that way. So both will actually get detected, both transversions and transitions. Okay, so again, this is hopefully not beating the dead horse too much here. Again, this is review from last time. What are our major problems? Major problems are we like temperature, we like oxygen, and unfortunately some people like smoking too, which often leads to this. So the main kinds of damage that you have in DNA have to do with what's going on with the individual bases, and to some extent what happens in the sugars and the phosphates. The size of the arrows here represents how common any particular change is. And if you look carefully, the big arrows, the ones I also put purple boxes around, are these here. So here you have blue arrow, meaning hydrolysis, and happening here for the two purines. So depurination. Depurination is the most common form of DNA damage, and it's just because we're in an aqueous environment, we like it hot, as it were. So depurination happens very commonly, and all that is hydrolysis of the glycosidic bond between the ribose and a purine. The next most common is this hydrolysis, which happens right here in cytosines, deamination of cytosine. And we'll see how this is happening again here as we move forward. Also oxidation, again, we like to live in an environment with oxygen. Particularly common is this position right here, the 8 position in guanine, and some of these double bonds as well in adenine. But hydrolysis, surprise, surprise, is the most common of these changes. So another way of looking at this is actually from our textbook, deamination of cytosine. You have here's cytosine. Again, it's a hydrolysis reaction whereby you lose this amino group and end up with this oxygen right here. Depurination, supposed to be shown here. Here's the ribose down here, your backbone. Hydrolysis, you lose the base and you end up with just an OH at that position on your ribose, no longer having a base there. This is, again, an overview of just one guanine oxidation, particularly here at the 8 position. You can also have alkylation, deamination of guanine, sort of you name it, all kinds of different possibilities here. The one that we haven't talked about yet is what happens particularly in the presence of UV light. These are so-called the cyclobutane rings, or a pyrimidine dimer. This pyrimidine dimer can happen between any pyrimidine, so it can happen between C and C, C and T, but most often people talk about these as thymidine, and these are the most common ones as a thymidine dimer. This cyclobutane ring, which forms right here, if you look at this, it's literally a rectangle with straight bonds. If you think about the way DNA is oriented, bases are offset from each other. It's what Watson and Crick came up with in their model. And we know that's true for most DNA most of the time. What happens if you have this now rectangular and planar bond between these bases is you have to twist the DNA to do that. So you really mess with the structure of DNA in order to get these pyrimidine dimers formation. Then the last one that I wanted to talk about is the DNA damage, per se, is this deamination reaction. Now, this is a very dangerous deamination reaction from the cell's point of view. We'll talk about 5-methylcytosine later, very common modification to DNA which happens. This can also be deaminated. This deamination, unlike leading to uracil, which is what you have with deamination of cytosine, leads you to thymidine. What's the big problem with this? It's a normal base. There's no way for the DNA repair machinery to recognize that this is an incorrect base, because chemically it's identical to thymidine. Yes?
What is it that causes there to be more thymidine dimers than
Okay, what is it that causes there to be more thymidine dimers as opposed to cytosine dimers or cytosine thymine? Has to do with the electrical structure of the bonds in the rings. And send me an email. I can send you all kinds of literature on this, but we don't need to get into details. This is not a chemistry class. Okay. So again, to hopefully avoid beating the dead horse here, main kinds of damage: cytosine deamination and depurination. So depurination, you have a guanine, hydrolysis, your depurinated sugar. Deamination, cytosine, gives you uracil. Or we also have our pyrimidine dimers, which have formed right here. And again, importantly, this is a planar structure, so it's really twisting the DNA to give you this kind of process. So how do cells deal with this? Lots of different ways. The smart cells, i.e. not us, because we've actually lost this activity. Quite why is not entirely clear. So-called photoreactivation. So photoreactivation is this wonderful DNA repair mechanism that recognizes when you have this pyrimidine dimer formation, which has happened, again, through the activity of ultraviolet radiation. There's this protein called DNA photolyase, which specifically binds to these pyrimidine dimers, and in the presence of a slightly longer wavelength light, reverses them and gives you back what you started with. E. coli's got this, Saccharomyces cerevisiae has this. For some reason, humans seem to have lost it. No quite obvious reason for that. Seems like this would be a much more efficient way than the way that we deal with pyrimidine dimers, which is a much more costly mechanism that we'll talk about in just a second here. So I mentioned mismatch repair. This is what happens usually right after replication has taken place, when somehow you've had either a transition or transversion. The wrong nucleotide gets put in. And sometimes that actually has to do with the tautomers. I don't remember if you all remember, if I didn't mention it particularly. When Watson and Crick were coming up with their structure, they said aminos instead of iminos. and ketos instead of enols for the individual bases? Well, these are tautomers. These are chemical compounds that will switch back and forth. And just very recently, people have found that a lot of this misincorporation really does have to do with the fact that you have one of these forms versus the other. Why does that make a difference? Because it changes the hydrogen bond formation. So you actually end up with the incorrect nucleotide, which gets added. And so this is almost always going to be transitions because purines are going to be similar, pyrimidines are going to be similar to each other. So it does happen quite a lot. So we had our fidelity that you have for replication. Fidelity that you have from replication is 1 in 10 to the 5 mismatches that the DNA polymerase makes, because when it folds down and checks that structure, it makes a mistake. 1 in 100 times of those mistakes, that gets repaired by proofreading. And then to get your numbers up to the final replication, so 1 in a billion, 1 in 10 billion, you have mismatch repair. So how do we know about mismatch repair? Well, a couple of things. The first way people knew about mismatch repair was genetics, studying E. coli and finding particular mutants in E. coli that incorporated a whole bunch more mutations. And so these are called the mutator genes. And so if we think about these particular genes in E. coli that lead to higher mutation rates, they're very often called MUT for mutation. So mutator genes in E. coli, what happens is there's an error which forms, again, because the DNA polymerase is put in the wrong nucleotide, the exonuclease hasn't gotten it. What happens? And this is recognized by the structure of the DNA. So both transitions and transversions are going to change that structure. Then you have this mut-S protein that binds to this mismatch. That's actually shown right here. This is the X-ray crystal structure of mut-S bound to a mismatched DNA. Then you have the mut-L protein, which will make a cut in the DNA on both sides of this particular change, or the incorrect, and then this gets repaired. What's the problem with this? You've just incorporated the incorrect nucleotide. You're going to cut out one strand and remake it. What if you cut out the strand that was the template strand and remake it? It's clearly going to be a problem because that's not the DNA sequence that you started with. So you have to have a way of recognizing what's a new strand and what's an old strand right after replication takes place. E. coli, it's easy. Why? Because you have methylation, which happens. Remember how E. coli knows that it's done replication from ORI-C? Once you've started replication, it's hemimethylated. So one strand has a methyl group on it, the other strand doesn't. Is it the new strand or the old strand that has methyl groups on it? It's the old strand, the template strand. So if E. coli sees we have a mismatch because the structure of the double strands are wrong, then mut-L is going to find this non-methylated DNA, cut it out, and we'll be good to go. What do we do in eukaryotes? Okay, we don't methylate our DNA, or at least not as much as bacteria do. Turns out that in eukaryotic DNA, as I mentioned really briefly, but it bears repeating again, the Okazaki fragments are a lot shorter in eukaryotic DNA than they are in bacterial DNA, only about 100 nucleotides. And so what that means, about every 100 nucleotides, you're going to have a little break in the new DNA on the lagging strand. So nicks on the lagging strand, easy to repair stuff on the lagging strand. What about the leading strand? Turns out that even on the leading strand, there's a bunch of nicks that happen, and that's because the polymerase comes on and off. Exactly how that works, researchers are still trying to figure out. So it seems to be already pre-existing breaks in the DNA, which are then recognized as being new DNA by the mismatch repair machinery in eukaryotic cells. And we mentioned last time some of the mutations that are present in a number of cancers also are involved in DNA repair processes. And one of those particular ones has to do with binding and recognizing NICs. This is this HNPCC. What was this again? I can't remember it. I can't expect you guys to remember it either, can I? This is the colon cancer gene. What was the first name for it? I'll remember later. But it's important for mismatch repair. So we have mismatch repair. We also have the other kinds of changes which happen. Which are most common? Which are the most common ones? Depurination and cytosine deamination. So the most common of these, not surprisingly, have their own mechanism for repairing these particular kinds of changes because they're really common. This is what's called the base excision repair process. Happens for deamination cytosine, but also for depurination. This is a very specific and very efficient process, but it depends on recognizing exactly that change. So, common changes. Here we have deaminated cytosine is a U. Okay, you shouldn't have U's in DNA. So there are sensor proteins, we'll take a look at them in just a second here, which recognize this U, and a uracil DNA glycosylase. So ACE enzyme, what does it cut? DNA, where there's uracil, it cuts the glycosidic bond. Again, this is between the sugar and the base. Then you have an AP DNA. What the heck is AP? Well, AP is apurinic or apyridimic. So it's just a DNA which is missing a base at that particular position. Then you have an endonuclease. What does an endonuclease do? It cuts in the middle of DNA, not at the ends. So that endonuclease will cut right next to where you're missing a base. There's a phosphodiesterase that will cut out one and only one nucleotide. And then DNA polymerase, DNA ligase will put this back together. So this is a great, very efficient process. You had one change up here, one thing happens down here at the bottom, but you have to have some specific way of recognizing either uracil as a change or your AP site as a change. On the other hand, if you have big changes that you don't know exactly what the problems are, there's a less efficient mechanism, but it's sort of the cleanup process. And this is what's called nucleotide excision repair, used in our cells because we don't have photolyase, for repairing things like pyrimidine dimers, and whenever there's sort of a big change, often a less common change, that's going to happen in your DNA. So here, instead of coming into this pyrimidine dimer and repairing it itself, there are endonucleases that will cut around where this change is, a DNA helicase that removes this piece of DNA, and now you have a template, you have a 3' end, and this can be polymerized and religated. But here, one small change, you've got to put in a whole bunch of extra nucleotides in order to repair this change. But instead of recognizing a single change here, now you're just recognizing something's wrong with this DNA here. Something's wrong, we need to take care of it. It's not very specific, again, uracil or one of these AP sites. So let's look at these in a little bit more detail. Here's the deamination process. Deamination can lead to all kinds of weird bases. We haven't really talked about adenine or guanine deamination, but important here is this uracil. And again, this is the most common of these. Cytosine deaminated to uracil. This needs to be detected. Well, how does this get detected? It turns out that, it would normally be stuck inside the DNA in the double helix, right? Well, it turns out, and this is still amazing to me, that there are what we call surveillance proteins that basically scan their way along all the DNA. So 6 billion base pairs in any of our given cells, and basically are checking every single base. Are you a uracil, you're a uracil, you're a uracil, you're a zero, blah, blah, blah. And in that process are literally flipping these bases out of your double strand in order to check and see if these are incorrect. So if there is a uracil here, the uracil DNA glycosylase will chop it off. Same thing is true for xanthine and hypoxanthine. Once that's done, of course, you have an AP endonuclease, and that can be repaired. So here's that process. And again, here's one of these proteins bound to one of those flipped out bases. Uracil, the DNA glycosylase cleaves it off. Now you have an AP site, an OH here. Again, this is the glycosylase which cleaves this off. The AP endonuclease will cut here. There's an either exonuclease or phosphodiesterase that chops this off. Now we have a 3' OH. DNA polymerase can extend this. Then you need a DNA ligase to put these guys back together. Sure, yeah. The enzyme that takes off the uracil is the uracil DNA glycosylase. Yeah, and this will be true for any of those particular changes. You've got a very specific change that happens. So, for instance, you also will have a xanthine DNA glycosylase, which will cut xanthine off of that particular place. So any of these DNA glycosylases are going to be very specific. Okay, how do we do nucleotide excision repair? This is different because it's not that individual nucleotide that you're scanning through the whole genome to check. Now you're just looking at the DNA to say, oh, here's a problem. I don't know what that problem is. Oops. So I just literally made a exonuclease here right now. So what's the problem which is going on with the DNA? It's a structural problem. So again, this should sound really familiar because that's exactly what the DNA polymerases are seeing. They're seeing the structure of DNA. So here that structure is recognized. In E. coli, it's recognized by a complex of proteins, again discovered genetically. How are they discovered? These were E. coli that were very sensitive to UV irradiation. What happens with UV irradiation? You get pyrimidine dimers. Change the structure of DNA. So UVR, these are now the UV resistance proteins, which once you break are now no longer resistant to UV, which will bind to this changed DNA. That changed DNA almost always gets bent, and you may have noticed also in those previous structures that we looked at, the DNA structure is very often bent where you have some of these changes, and that seems to really help all of the processes of cutting, religating, etc. So you've bent the DNA, now you have nicks that are going to form over. So when I say nick, people always ask me what do I mean by a nick. Nick is just literally cutting the phosphodiester backbone by hydrolysis of one bond. And that's almost always between the phosphorus and the OH on the sugar backbone. So it's just a cut of that one bond. You're not missing a nucleotide at that point. So you actually are always going to need either a nick and an exonuclease to chew off a nucleotide, or an apurinic site, or you'll have a nick somewhere else further along, as you have with nucleotide excision repair, where you have a whole piece which gets removed. So DNA polymerases can't do anything with a nick. They've got to have a full template in order to put in the correct nucleotide. So here we have two nicks that are put in either side of where you have this change, a helicase which removes this piece of DNA. Now you have a 3' OH, a template, DNA polymerases come along, and then this DNA polymerase is eventually going to run into this end of DNA where you just remove this. This now gets put together. Sort of the opposite of the nick and the nicking activity is now DNA ligase, is putting those two back together. So you have a nick in the absence of any other DNA damage. A ligase can fix it. If you have a nick and then you've started to remove nucleotides, either one in the case of base excision repair or multiple ones in the case of nucleotide excision repair, then the DNA polymerase has to come in, and eventually you're going to have a nick that needs to be repaired. Yes?
So notice on
So the question here, ATP dependence of the whole process. So the main ATP dependence of all of these reactions is just like you have in replication, is with the helicase activity. So anytime you talk about nucleotide excision repair, you've got a piece that have to be peeled off. So the nucleotide excision repair always requires ATPase. In the case of base excision repair, you're going to have hydrolysis of the backbone, but that's going to be an energetically favorable reaction because you're increasing entropy in the process. You don't actually need ATP for those. Now, you of course need nucleotide triphosphates for the enzymes in order to fill stuff back in and also for the ligase, but not for the individual glycosylase. Okay, we're all happy? Answered our questions? Good. Everyone's ready for their clickers? The mechanism of mismatch repair, sorry, that's AIR is missing there, is most similar to the mechanism of photoreactivation, nucleotide excision repair, base excision repair, proofreading, or chromatin remodeling. What was that? That was the last exam, right? Now, is most similar to, sorry that got chopped off there. Ten. 1. Haven't looked at this. Okay, we've got a distribution here of possibilities. Nobody likes my chromatin remodeling. Okay, that's fine. I have no problem with that. Photoreactivation is through what? The photolyase. So nucleotide excision repair, base excision repair, or proofreading. Proofreading is just exonuclease activity of one nucleotide. Mismatch repair, how does that happen? You've got a change which is detected by a structural difference in your DNA. That change in structural difference needs to nix on either side of that particular change. It gets removed by a helicase and then refilled in. What's that most similar to? Nucleotide excision repair. Exactly the same process, nix on either side, filled back in. Is that clear? Yes? Not? More questions on this? Okay, so a couple of other things that I wanted to mention just as far as DNA repair is concerned. And we'll talk more about this as we talk about transcription later on. It makes perfect sense that you would want to repair any kind of DNA damage in parts of your DNA that are actively being made into proteins or actively made into RNAs that you need for, say, your ribosome or some kind of regulatory function. So it's been really quite well shown that a lot of these DNA repair proteins, such as the UVR A and B right here, are actually associated with the RNA polymerase. The RNA polymerase, again, is what's transcribing. That brings repair proteins along with it. And so what you see, and this has been shown also in eukaryotic cells, and we'll see how that happens later, that the DNA repair machinery is with the polymerase as it's transcribing any particular piece of DNA. So if you look at genes that are being actively transcribed, so this would be in eukaryotes in the U chromatin, those get repaired much faster than any kinds of changes, mutations that have happened in heterochromatin. And the reason for that, they're associated with the RNA polymerase. It's bringing them along. So this is just an example of that here. So those are the, oh, sorry, Diana. So the question is, does the switch here between the polymerization, which is happening, transcription happening with your RNA polymerase, and displacing this and getting these to start? Again, this is always going to be your ATP hydrolysis switches here, which is going to be involved not so much in the binding, but the release of these two proteins, the UVRAs from the UVRB. But I would say in terms of ATP hydrolysis, getting back to the question over here, it's really about the process. And here, again, UVRA and B are involved in which process? The nucleotide excision repair. So you're going to have a big piece that needs to be repaired. So that process is highly ATP dependent. Yeah? Mm-hmm. Okay, yeah, so the question here, and sorry, paraphrase, correct me if I'm getting it wrong here, is basically the RNA polymerase itself detecting problems. And the answer to that is basically yes, because the RNA polymerase, just like the DNA polymerase, is detecting the structure of the base pair. And if it gets somewhere, it's like, oh, wait a minute, can't detect this anymore. That mostly seems to be that the polymerase stops or stalls, and at that point, the repair machinery can get onto the DNA and repair it. So the question now is, does the polymerase then dissociate and reassociate at that position, or does it stop and then restart? The answer is yes. It does both. Okay, other questions about these individual nucleotide kinds of changes. So all of these are dependent on having that other strand be there, right? Because that's where your template is. This is how you can be repairing. What happens if you have a double-stranded break? What happens if you have both of the strands that break? This is really a problem because you don't have that opposite strand there anymore. So how is that dealt with? Well, first, how do you get it? We talked about Fukushima last time, but it turns out radiation therapy, radiation oncology. What do? They zap the tumor with ionizing radiation to generate double-stranded breaks. A number of chemotherapy drugs are important for generating double-stranded breaks. Double-stranded breaks are a real problem for the cell. But they still happen, and we'll talk about probably the main way that they happen in just a second here. It has to do with replication. But clearly a big problem. How does this get dealt with? One of the ways that it's dealt with is the quick and dirty way, which is non-homologous end joining. You got a double-stranded break, 2 bits of DNA floating around, just put them back together. Which is, again, quick, but also really dirty, because in that process, as we'll see, you have to lose a couple of nucleotides. And if that happens to be in a coding gene, we've got potentially real problems there. Again, some people think this is why there's so much of our genome that's non-coding, so that you can get away with non-homologous end joining and it not be too dangerous most of the time. On the other hand, the much more energetically costly way of doing this, but also a much more accurate way of doing this, is what's called homologous recombination. And homologous recombination depends on having some homologous DNA that you can then use to repair that break. And the main thing is figuring out where those two ends should end up relative to each other. And this is great in diploid organisms like us, because you've got mom's chromosome over there, even when dad's chromosome has gotten broken up. So you can use that as a template for this repair. Turns out that most bacteria are undergoing very rapid replication. So you often have 2 copies of the genome at any given time. And that's how homologous recombination seems to work in bacteria. So let's look at this again. Cartoon form, non-homologous end joining. You have a double-stranded break. This gets chewed in and put back together. Quick, but again, dirty and potentially rather dangerous. On the other hand, homologous recombination, you have a double-stranded break. And as we'll see, you actually lose a couple of nucleotides at the end here as well. and that's partly because these double-stranded breaks are rarely right opposite each other. And one of the first things that happens is you have what's called end processing, which will generate a loss of nucleotides there. But if you have a homologous chromosome here, and in this case just shown as the sister chromatids, it can use the information from this chromosome to repair the information which was lost over here. So let's look at these processes. Non-homologous end joining, again, the quick and dirty process. There's a double-stranded break that happens in the DNA. This is recognized by a particular protein. Kind of a fun story with this protein. It won't be on the exam, but one of the guys who discovered it, I bought a stereo from in graduate school. So it binds to the ends of DNA, this DNA protein kinase, which will phosphorylate these things, a whole bunch of additional proteins. Additional proteins means you don't need to know their names for exams, that then chew in the DNA and pop them back together. So this works. You get ligation, but you almost always lose a few nucleotides in the process. Again, this may seem, really inefficient, but again, most of our genome is not coding, and so losing a few nucleotides here and there might be okay. But it turns out that this process is also absolutely critical for adaptive immunity in vertebrates. You hopefully know that antibodies have a huge amounts of variability in them. A lot of that variability actually comes from chopping the DNA and putting it back together in extremely similar mechanisms to this non-homologous end joining. So generating antibody diversity is apparently taken this particular mechanism and now used it to generate diversity. Usually, diversity is a bad thing, but if you're talking about antibodies, diversity is potentially a very good thing. So that's the process of a non-homologous end joining. Let's talk about homologous recombination. This is a much more involved process, but again, it's a much safer process in terms of keeping that genetic information, or at least finding a copy to repair that genetic information wherever it was. So this is the overall process, and we'll go through each of these steps here in a little bit more detail through the rest of class. Start out with one chromosome, you have a double-stranded break. The first thing that happens after this double-stranded break is 5' ends are degraded by an exonuclease. So this exonuclease chews in at 5' ends, leaving this 3' overhang. And it's both ends this happens to, so you're going to have a 3' overhang here and a 3' overhang here. This 3' overhang, single-stranded DNA, then does something called strand invasion. So here's this single-stranded DNA goes and finds a homologous chromosome and will now bind to one of those strands and displace this other strand. So this now is going to form what's called a branched DNA, because this is your branch right here, and this branch moves along the DNA due to DNA replication here, because you've got a 3' OH, you have a template, this moves along. You notice now this maternal strand here is eventually now going to get to this 3' overhang, which can now base pair, and now you've got another template. DNA polymerase is perfectly happy, it can extend here, but now you need to do something about the fact that these two chromosomes are now crossed over and linked to each other. There's one strand which is getting passed from one to the other. That's what's called resolution, and it is by cutting the two strands and putting them back together. In that process, sorry, back up a little bit. This crossed over form is called a holiday junction right here, and the resolution of these holiday junctions is cutting the DNA and putting it back together. And we'll look at those again in a little bit more detail as we move through here. The first step is strand invasion. So you've got a 3' overhang, single-stranded DNA. That's bound to by a particular protein. It's called REC-A for recombination in E. coli. It's a homologous protein in eukaryotic systems and in archaeal systems. What it does, it binds to this single-stranded DNA and basically helps that single-stranded DNA to now go into a duplex DNA and exchange these two strands. So here's your normal DNA. This is going to be your, well, let's say the paternal chromosome got chopped up here. Here's your maternal chromosome. Rec-A-bound form in the presence of ATP now can get a heteroduplex to form. ATP hydrolysis happens, and you end up with double strands from this invading strand and one of the two strands that you had in the first place. So this DNA invasion process also, hang on, shown, no, I think I've skipped this, got out of order here. Oh, this DNA invasion process, dependent on Rec-A and ATP, the ATP hydrolysis basically seems to be getting rid of the Rec-A and giving us our double-stranded DNA, also known as a heteroduplex. Why is it called a heteroduplex? Because you've got one strand which has come from paternal, the other strand which has come from their maternal chromosome. Now, this will be your first step, and then the DNA polymerase can extend this, etc. But one question that, was always a question for me to start with is, this is great if you've got maternal and paternal. What if you're a poor E. coli? And how do you then deal with this? And how do you get double-stranded breaks in the first place? Turns out that a lot of double-stranded breaks have nothing to do with ionizing radiation, which is probably a good thing. We're not exposed to ionizing radiation most of the time. But lots of times we have breaks in our DNA. And if there's a break in the DNA, if a replication fork gets to that, so here it's going to be a break in your DNA on the template of the leading strand, the DNA polymerase is going to get to here and the helicase, and now, if this has not been repaired, now you have a double-stranded break. Just the fact that you've been replicating through a nick on a template strand, you're going to end up with a double-stranded break. Now, the nice thing about this is you also have your lagging strand right there. So this is automatically a template for homologous recombination. So here, double-stranded break, exonuclease degrade 5' end. Rec-A will bind to this DNA, which is a match. Here, you have strand invasion. Now you've got a DNA polymerase which can extend right here. Down here, normal lagging strand replication can take place, your primase, your Okazaki fragments, etc. Then all you need to do is to resolve this holiday junction, and you're happy in terms of your replication fork. This is probably how the vast majority of double-stranded breaks happen, is just in replication. Because DNA makes nicks, as we saw. DNA is very fragile. Even this model up here is fragile. So you have lots and lots of these double-stranded breaks. Turns out that double-stranded breaks, because you can do this swapping over of genetic information back and forth, probably most of you, oh, sorry, yeah.
Oh, I'm sorry. I think I'm just missing something. Are those just something that happen, or is that from a one or two making a nick and then not, putting it back together normally?
Okay, so the basic question here, again, correct me if I'm misunderstanding you here, is that where do those nicks come from? There's basically, the nicks seem to be either the DNA ligase hasn't gotten around to fixing that particular nick, or it's a hydrolysis which happens. Hydrolysis happens in the backbone of DNA. So nicks happen, basically, I think is the point as far as that's concerned. Now, there's a slight difference. So nicks happen, yes. But if you think about, hopefully, remembering way back when to principles of biology, you remember what happens during meiosis when maternal and paternal chromosomes get together? They do all this crazy, nasty crossing over stuff. Yeah, that we're supposed to put black boxes over. So it turns out that this, why do we have sex in the first place? a lot of that is because you can repair alleles. Now remember, this is if you don't have a recessive allele that's potentially a real problem in your population, how do you get rid of it? You get rid of it by grabbing the genetic information from the other strand. How does it happen? Meiotic recombination, crossing over. This seems to be, as far as I can tell, I need to go back and check the literature again on this, but this kind of crossing over of chromatids that happens during meiosis seems to be pretty required. So you almost always have this crossing over that takes place. And it's only relatively recently we figured out how that happened. It turns out the way that happens is through double-stranded breaks. But it's not a double-stranded break because of a nick that just happens to be there. There are specific proteins which will bind to DNA in meiosis and make a double-stranded break, so that you can have this homologous recombination that takes place. Yeah?
So on the holiday junctions where they're binding together when they're the two strands, is that similar to the chiasmata function in crossing over during meiosis?
Okay, so the question here is holiday junctions versus chiasmata. So the chiasmata is literally cytologically, looking under the microscope where you see this crossing over that takes place. That is at the gross cytological level, just microscopically where they're crossing over. The molecular level, that's where you have these holiday junctions, which are right here. But underlying all those chiasmata are going to be these holiday junctions. Yeah? So REC-A is specific to bacteria. There are these proteins called RAD proteins that you see in eukaryotes, RAD51, RAD53, which have practically identical structures to RAD-A. REC-A, excuse me. There's also a RAD-A which is present in archaea. But they're very, very similar structurally and practically identical functionally. Now, and you're exactly right in terms of this example. We're talking about meiosis. Meiosis doesn't happen in bacteria. So these are specifically going to be eukaryotic proteins that are involved here. And so during meiosis, double-stranded breaks are generated. Then you have this REC-A type, which is why I put REC-A type here, because it's RAD51, which leads to strand exchange, DNA synthesis, and religation, and formation of these holiday junctions. So just getting back to your question about the chiasmata, that would be these two, and of course it's many more than just two, but example of two of these homologous chromosomes, maternal, paternal. They're going to have lots of examples of this either crossing over, where you exchange one bit of chromosome here, but also many regions where just a small piece has been exchanged in terms of genetic information from one of these chromosomes to the other. So how do you get some cases where you have complete crossing over and some where you have just a little bit which has been exchanged? So this is this process, and we'll spend basically the rest of today talking about this because it seems to be a complicated concept to think about. And it has to do with the fact that we always draw DNA here with two lines next to each other and they're actually really wrapped around each other. So we'll get to that in just a second here. So if you think about meiotic recombination, or for that matter, double-stranded break repair, you'll have these holiday junctions which form because one of these strands is invaded, this strand is copied, you ligate this end, now you have an X structure, this end gets ligated to that end. You're always, if you have double-stranded break repair, going to have two of these crossed over pieces of DNA, holiday junctions, not Christmas or Easter or something like that. That's from Robin Holliday, who predicted these, and these have been shown since then to actually be the case. When you have holiday junctions, there are basically two ways that you can cut and religate these. And these are shown here as either the horizontal arrows or the vertical arrows here. These, of course, look completely different relative to each other. Now, this doesn't look like a cutting and religating that's the same as cutting and religating here. But if you think about the double strands wrapped around each other, it really does. It's a completely symmetrical process. And what happens is if you have resolution of these holiday junctions, here, if we just think about these horizontal ones, it's going to be orange going to red here, and red going to orange down here, red to orange, orange to red. That gives us a small piece of DNA right here in the middle where you haven't had what's called a crossover. What do you mean by crossover? It means that genetic information here at the ends is the same as what we started with. So here, orange at the top, red on the bottom, orange at the top, red on the bottom, orange at the top, red on the bottom, orange at the top, red on the bottom. If, however, you have different, either horizontal or vertical cutting here, what ends up with is you end up taking this strand right here, this orange one, and hooking it up to the red one down here. So orange here, cut and ligated to red here. That process will give you this crossing over. So now we started out with orange up here at this end and going off, of course, to the left-hand side here. Now we've got orange down here. So that's your classic crossing over and your chiasmata which have happened here. So that's your crossing over process. What do we mean by this? Oh, this is the slide I was looking for before. Your question about RAD-A, Rec-A. So RAD51 is the eukaryotic, and when we say eukaryotic here, we really mean the yeast equivalent, which binds to single-stranded DNA and allows this strand exchange to take place, again, in the presence of ATP with this process. Okay. Yes, over here. Yeah. Sorry, I missed the last part of your question. Okay, so the question here basically has to do with these end pieces. These are supposed to be diagrams of little pieces of your chromosome right in the middle. So yes, you could have exchanged the very ends, but this can happen anywhere along your particular chromosome. And particularly if you're thinking about meiotic recombination, that's going to happen all over the place. And so maybe back up here really quickly. Here, each time when you've got red and blue that have been exchanged, that's where you're going to have either crossing over, which takes place. So here you've got the blue at this end and the red at that end. But lots of non-crossovers, each of these vertical lines that are present in the rest of your chromosome. Okay, so finish up with a clicker question. Yes, no, Either that or I can talk for another 20 minutes. No. So after a double-stranded break in DNA, what's required before a strand invasion can take place? Again, a review of all of these things. And maybe I can get this to actually start. That would help. Start? Yay. 5' to 3' endonuclease, 3' to 5' endonuclease, 5' to 3' exonuclease, 3' to 5' exonuclease, or holiday junction resolution. Ten, 5, we all happy with that? 5' to 3', so am I. Good. Okay. We will look at holiday junctions next time. All of your scantrons are here alphabetically by last name.